So we want to be able to focus our eyes. So our eyes will diverge and converge depending on what we need to do. So if you are bringing something into the center, they're going to converge so that you can see better. We adjust the eye by muscles to make sure that they work, and we also adjust the lens so that we get the best vision possible. So there's a lot of area in the brain that's related to the eyes because we have so many receptors and we do so many things with our eyes. They're so very important to us for survival even. Imagine if a caveman went blind. What would they do? They'd die because they couldn't eat, they couldn't hunt, they couldn't do anything. They wouldn't let, other people can't just lead them around all day long and bring them food, right? So they just put them out and let them die. So close vision requires accommodation. The changing of the lens, the refractory power of that image. So we have what they call presbyopia, which is a loss of accommodation of eyes. These are old timers' eyes. When people start getting bifocals and trifocals, that's presbyopia. You can't see the way they should. If people start holding their things way out here to read them, that's presbyopia. They've lost that close vision that they need. Right. So we can do this by constriction of the pupillary reflex, bringing the eyes together. We can converge the eyes, I should say rotate the eyeballs towards each other, and we can constrict them where we change the shape of the lens and the muscle. So everything works the same way. This is parasympathetic, the same way. It changes the shape of the eye a little bit, but we still get the impulses to the back of the eye so we can see properly. Now, we have different things that people are. How many people are nearsighted? Okay, that means that the focal point is in front of the retina and your eye is longer than normal eyeball. So you have, if you have glasses or contacts, they are concave lenses. It changes the way they are. So nearsighted, then we have hyperopia, which is farsightedness. This is a focal point is behind the retina, and I'll show you what that means in a minute here. And we give convex lenses. Some people have these huge concave lenses and you can actually see where they're indented and they're pretty thick. And others have these really thick ones in the middle and thin on the edges because they're almost blind. Does anybody have really bad eyes in here? Well, that's right. You said you had really bad eyes. So are your glasses thick? Or nowadays they're not quite so bad, but in the old days, the glasses got really thick, like pop bottle thick. They were so heavy, you're always pushing them up, and it hurt the back of your ears, and it was a mess. Then they came out with plastic ones, and it's like, whoa, so much lighter, so much better. Stigmatism is caused by unequal curvatures. They're almost like they're football shaped. They're not cylindrical. They're not round eyeballs.
So they say I have astigmatism, but like my vision is different in each eye.
Yep. Yeah, one eye that's shaped differently than the other one probably. So they have to counteract that by proper lenses. So this is emmetropic. This is normal. So the impulses are coming in, right? We got these light impulses are coming in. They hit the lens, and they converge at one point in the back of the eye. Pinpoint, right there. That's emmetropic. That's perfect eyesight, 20/20. Let's see, how many people, how many people wear contacts or glasses in here? Anybody that doesn't? Tevin's the only one that has eyes that you don't have corrected? Wow, that's pretty remarkable. That's good. Keep it that way. Now this is myopic. This is nearsighted. So this is normal at the top, where you have the eyes. Well, it's not normal here, but this is what it would come to the back of the eye if it was normal. But here, the eyeball is too long. It's like it's stretched out. So the image now, with the lens the way it is, falls short from being pinpointed in the retina. that point of light doesn't hit the back of the retina properly. So what we do is we put a lens in here. We correct it. We divert that light just a little bit by looking at the corrective lens and the lens of our eye. Knowing the shape of the eye, we can pinpoint that image to the back of the retina again. And then we can see it crystal clear. If you go to the doctor's office, the eye doctor, right, what do? Sit you in the chair, they put this thing in front of your eye. What do they ask you? A or B? This one or that one, right? You go, come on, I don't want to do this. They look both pretty good, right? Both of them.
They all look the same, honestly.
Yeah, they get to the point where, I don't care, just let's go on, you know, instead of bagging it back and forth. But they're trying to make it so that this lens fits perfectly with your lens so that that image goes right to the back of the retina, pinpoints right on that retina, because if it's too short or too long, it causes a fuzzy image. So he'll say, this one, nope, that one's fuzzy. How about this one? That's better.
So why have they done that to me if I've got good vision?
Why haven't they done that to you?
Or why have they?
Because they want to make sure you still have good vision. Right? You could have, your vision could be changing slightly. You may not need glasses quite yet, but it could be that way. This is hyperopic eyesight, farsighted, where the image would actually line up at the, not at the back because the eye is too short. So instead of being crystal clear, sharp image, it hits, and it's a wide image on the light. Too many receptors, too many impulses hitting at one time, sending images to the brain's confused, it looks all blurry. Looks like a mess, right? So we put a convex lens in front of it. We bring that image down, that light source down, and then focus it to the back of the eye, just like we did with the others. So rods and cones. Rods and cones are the receptors in the back of the eye. They are help us to see visual images. The rods are more for darkness. The other ones are more for light and images. Put your cell phone away, please. So we have, those images are constantly absorbing the different light sources, the different amounts of light. This is where they come. So this is the inside of our eye. This would be this is how the vitreous humor. The light would hit this, and then it goes from here all the way through to the back of the retina. From there it goes out to the rest of the body. Right? To the rest of the head, I should say, to the brain. So the rods are more sensitive to dim light. Let's see if we can get this YouTube video to work. It's sometimes they get mixed up. Oops, that didn't work. Let's do it on here. It should be focusing in the right spot in the back of the eye. It makes everything work the way it should. Come on, YouTube, let's go.
17-year-old Corey Hanna has always seen the world a little differently than you and me. He has night blindness, so when the sun goes down, Corey's world goes from this to this.
Most of the time at night, I don't go outside of the court.
It's not such a big deal when it's summertime and it's light till 9, but now it's light at 5, or dark at 5:30. It's an issue.
Forcing Corey to spend his evenings indoors where he can see. Doctors say there are two kinds of cells in our eyes, rods and cones. Cones help us see in the bright light, rods help us see in the dark. But for some reason, in Corey's case, his rod cells are dying.
When that happens, someone has night blindness. But then unfortunately, about 5 or 10 years later, often the cones die.
Stuart Mengel is a researcher at Ohio State University Medical Center. He says even though they play separate roles, rod and cone cells are connected by tiny channels in our eyes. Scientists never thought that made a difference until now. Using night vision goggles, Dr. Mengel and his team used goldfish to gain new insight into our eyesight. They injected glowing or tracer molecules into cone cells in the retina of the fish. During the day, the tracers stayed in the cone cells.
But at nighttime, the tracer went to 100 other cones and to about 1,200 rods. So it was a very dramatic difference, much more than we ever had expected.
That means for the first time, researchers have determined that rods and cones work together in the dark. And since the eyes of goldfish are so similar to ours, it could someday mean new treatments and a whole new world of night activities for people like Corey. At Ohio State University Medical Center, this is Clark Powell reporting.
That would be horrible.
Couldn't see at night. Couldn't go out at night. You'd be like you're blind. You couldn't register. Now you could see colors if it was a bright light. If it was somewhere like Las Vegas where there's lots of lights, that'd be okay. But in other words, you couldn't. So the rods are more for night vision. Gray tones, you find this a lot more in animals than you do in humans. They're more dominant. They'll give you more of a fuzzy, indistinct image instead of really crystal clear. That's what the cones do. They give us the crystal clear images that we have. So the cones need bright light. They have low activity, low sensitivity to low light. You can't use them at night too much. Just now they're saying that they work together, but we still have more of a better use for those during the light times of the day when it's really bright out. You have 3 pigments that could be used for colors. What colors do we normally see in our eyes?
Red, blue, yellow.
How about green? What do you get when you mix blue and green, blue and yellow together?
Green.
Green. So red, green, and blue, right? Those are our 3 colors of cones. But how do we get things like browns or pinks or green? We only have 3 different colors of cones.
By blend together.
By blending them. They all work together. So you have the blues and the reds are registering and the brain says, oh yeah, okay, that's green. I learned that. I know what that is. When you're a kid, do you know what your colors are? When you're 1 year old and your mom asks you, what color is this? That's all you say.
So are black and white colors?
Black is devoid of reflection. White is reflection of all the light off. Right? So when we look at the chemistry of our eyes, the visual pigments, we have this called retinol. Retinol is synthesized from vitamin A. So if you don't have vitamin A in your diet, you can go blind. These little kids in Africa and some of these third world countries don't have good diets. And they start going blind. You give them some vitamin A and it's like, whoa, I can see again. It's just phenomenal response. So it happens that way. And what it does is it combines this, it changes the eyes, it starts the action potential. The retinol converts the 11-cis retinol to all trans retinol, and this initiates the chain of reactions that leads to the action potential going down the optic nerve. Okay? So this is kind of how it looks like. We have this rod disc that's kind of inside this membrane. It allows for the opening of channels just like we do in any nervous system receptors. Works the same way. But we have to open up these channels, allow the chemicals reaction to take place. It elicits the reaction down through the membrane into the nerve and then into the optic nerve back to the eye. So rhodopsin is the big contributor to this. It's a visual pigment in the rods. And that's we have opsin and rhodopsin, and those are the ones that give us sight. Now, excitation of cones is different. They have 3 types of cones. We talked about blue, green, red. And the color blindness is due to a congenital lack of one of those colors, those cones. Now they've got new glasses now. I'm sure maybe you saw it on Facebook or something where they give this guy, he starts crying because he's never seen the colors before. Pretty awesome, I think. Gives him some sight. But they're doing a lot of good things. They're having corneal transplants. For your hearing, they can put in an implant so you can hear. Pretty good stuff. They're able to connect with nerves like they have never done before. So we're getting there. Light adaptation. This is when we curve from darkness into bright light. So if I turn off all lights, blocked off all the lights coming in, and we made this pitch black, and then all of a sudden you step out into the bright sunlight, what does your eyes do? They constrict, right? Your pupils will slowly constrict down so that you don't have much impulse coming in at all. And that takes about 5 to 10 minutes to physically do that, which isn't much time at all to get down to that point where you can adapt. Now your eyes are going to change really quickly, but the way that it works is too much sunlight in there causes too much reaction and almost blinds you. It creates too much impulse to the back of the head, to the occipital area, and we can't see that at all. So we have to let this all take place. Dark adaptation is going from bright light to darkness. This takes 20 to 30 minutes. The pupils dilate, the cones aren't functioning as much, but the rods are really functioning the most. Right? So, why do pirates wear eye patches? Do you know?
So that they can, so when they're up on deck, they can see in the lights through the one eye without the patch. But then when they go below deck, they take the patch off so they can see.
Did I tell you guys this already? Did I tell you this already, or did you just darn smart, right?
Just
Okay.
that's smart.
I thought it was just because the pirates had that little thing on their shoulder, that bird, and it picked their eyeball out, right?
I thought it would have been because of a lazy eye.
Lazy eye, or maybe he said he got stabbed by another swashbuckler. So the visual pathway, the axons from the retina ganglia form the optic nerve. So the retina is a sheet of receptors, and it takes the light in. They open up the channels. The receptors work just like they do any other nerve receptor, and it sends an impulse towards the optic nerve. The medial fibers of the optic nerve change. They desiccate. They cross right behind the eyes as it comes out through that area of the brain, the sphenoid, comes behind the sphenoid and it crosses right behind that. And what's from the right eye goes to the left side of the brain, left eye goes to the right side of the brain, and also fibers, the ipsilateral fibers go from right side to right side, left side to left side. So we have this ability to send the information. A lot of it first goes to the lateral geniculate body of the thalamus, and then from there out. Goes to the primary visual cortex, and then it ends up in the superior colliculi, and this is initiating the visual reflexes, what's going on. So we have lots of visual reflexes. Something comes towards your eye, you close your eyes, you blink, right? Too much bright light, your pupils dilate or constrict. So we have a lot of these reflexes, but they have to be stimulated by some type of a stimulus from the outside. Small cells in the retina contain the timer for our daily biorhythms. Right? So we have kind of this built-in time to go to sleep type of thing, or time to wake up. Right? So you're laying in bed on Saturday, and the windows shades are open. It's tough to sleep past daylight. When the sun comes up, you start to wake up. You don't sleep as well. When it's dark out, you go right to sleep. Not a problem, because that's part of our biorhythms. We're supposed to sleep at night and be awake during the day. That's the way we're put together. Then we have something called peripheral vision. So this is great for people that, if you have kids or you're a teacher, you can see a lot of things happening at one time. You see, I can be looking this way and I can see people texting off to the side, and so then I can shoot them with a whatever. glance, but you can get this to work. So we talked about the chiasma, how the impulses go from right side to left side, back and forth, and also from one side to the other. So we have this ability to get information from both eyes to the different parts of the brain. Now, one thing that we have is really kind of interesting is depth perception. We can take and we know how far it is to pick up something. We can take our hand and pick it up because we can stop right when we want to. It's like one of those little cranes at the stores where you crank them down and it picks them up. You know, we can stop where we want to. But other animals can't do that. They don't have that three-dimensional patterning. They see things in two dimensionals. So it's more difficult for them to do those types of things. So we have the lateral nucleus of the thalamus. It helps us to emphasize visual inputs. We get high cone density, and it tells us what's going on. Very sharp perception. We can see something that's bright and colorful, or we can see if the fruit is ripe or green. We know that those are good things. Cortical processing, we process all this information that comes into our brain. The outside of our brain takes this information and says, aha, I remember that, or this is something new, what is this? And we start to imprint on our brain what those images are. You know, how many people have ever seen a Tasmanian devil in person? Right? We don't know what they are. If you lived in New Zealand, that would be something that you'd see, or Tasmania. You'd see those, right? They may not know what a skunk looks like, but we do, because we've seen them. So those are some things that we imprint on our brain, and we see those images, and we remember them. Color, the motion, all those things come into play when we're looking at how this works. The temporal lobe processes identification of objects. Right? So if you have your favorite toy, that's you remember that. You'll remember it all your life. If you had a little teddy bear when you were a kid, it's stuck in the back of your brain. It's there. The parietal cortex and the post-central gyrus are there for spatial location. Where are you at in space? How far do you have to go to push a button without going through it or not touching it at all? That spatial relationships is really important for us. Okay? So that's kind of this eyes in a nutshell. It's kind of enough about eyes. Smell. Some people smell more than others, right? Some people are mixed up. Their nose runs and their feet smell. Ha ha ha. Some people smile. Okay, you're awake. Good. Taste and smell of olfaction. These go together very well. Taste and smell are really closely related, right? Chemoreceptors respond to chemicals in aqueous solution. So in order for you to taste something, you have to have a chemical that can be released into your saliva, and then the receptors on your tongue can pick it up. Same way with smell. You can't smell something that's not emitting some type of a chemical signal. Some type of a chemical is coming off of that. So if my iPad doesn't smell, there's no smell to it, no chemicals coming off of it, that's great. But if you have somebody that's sitting next to you and they just pass gas and the methane is all over the place, you're going to smell that, right? So smell is important for us. We need to have smell. Smell helps us to keep from picking the wrong fruit or dating the wrong person or picking up the wrong kitty cat, thinking that your skunk is a cat. You know, it prevents us from doing those stupid things. So smell is good. It also makes it really handy when we want to taste pizza and your favorite food, like lasagna. Right? So the organ of smell is your nose. The roof of your nasal cavity, there are little receptors hanging down inside your nose. So if you're going in and digging for gold, don't go too far, because you can actually damage the receptors inside your nose. You know, and no scratching it out with a putty knife or something like that, trying to get the boogies out. Okay. So they are bipolar neurons. That has nothing to do with that they're crazy or anything. They have axons coming off in two different directions. That's why we call them bipolar. The olfactory nerve is where all this information goes, and that's cranial nerve number 1. Goes to the smell. So the smell comes in, wafts up to the top, to the very back of the nose, past the inferior nasal conch, the middle nasal conch, the superior nasal conch, into the ethmoid bone, and there's where your receptors are, way up on top there.
So how does taste work then?
Taste and is on your tongue. It's all on your taste buds. And the nerves come together and kind of in the back of the head, well, not the back of the head, in the head, and creates that memory for taste and smell. It's just like the other day in lab. Did everybody remember smelling things, right? Could you have smelled and smelled things better if I would have gave you a picture? If I would take a bunch of orange cream or something, orange scents, and put it on a picture of an orange, that really makes it clear, right, what it is. But if I would take that same smell and just slather it on a piece of paper and let you smell it, would you have gotten it as easily? No. We put many of our senses together, and it gives us a overall picture. Yes?
So, what was the percentage of smell that contributes to tasting things?
70%.
70%. So, if we did get rid of taste, then we could still taste things through smelling.
Well, we can smell them, yeah, but you're not going to taste them as well. The smell dictates a lot of your taste, so your taste would be off. So it may not taste like it used to. You get ready to smell, because part goes to the same part of your brain. So you're only getting one part of your impulse going to your brain, not the whole picture. So it's going to say, well, I know what this taste is, but I just can't remember. So this is here's the bottom of your nose, or actually the top of your roof of your nose. It hits these little receptors, sends impulses up by way of these little ganglion, these little cells, up into what they call these microcells. And the microcells, they converge on the microcells, and then it creates a massive action potential that goes back to the olfactory tract into the brain through the ethmoid bone. So dissolved deodorants is what we have to have, and not deodorant like you put in your armpit, but anything that emits a chemical smell. Chemical comes off of it. It could be anything. It could be sweet smell, sour smell, but it has to have molecules coming off of the material that you're trying to smell. Now, dogs can smell, cats can smell better than we can. You know, we have a very limited range of smell. So, but the sodium and calcium channels are what's important for us. We have the sodium, and the calcium opens up the gated channels, and then the sodium potassium expand back and forth, and we get that action potential by way of depolarization and polarization. Okay, one video and then let's go. Okay, let's do this one here. This might be a little eye-opening for some guys, if it ever comes up.
The whole industry has developed around the idea that certain scents drive us wild and bring the possibility of sex one step closer. Scientists have long researched for a human pheromone, a secretion that influences sexual behavior. So far, they found only one that comes close. Men produce the hormone androstenone in their apocrine glands, which cluster wherever there's body hair. But it's hardly a love drug.
Females hate the smell generally of males. Androstenone seems to smell like musk in the beginning and then turns into a urine-like smell.
In Vienna, Austria, evolutionary biologist Karl Grammer is leading the world in the hunt for human pheromones. Early in his research, Grammer discovered that there was a point in the female menstrual cycle when women stopped finding the acrid smell of androstenone distasteful and began to like it.
Certainly at the point of ovulation, males smell good, and attractive males especially smell good.
So for men, androstenone is an efficient mating radar, repelling women unless they're fertile. But the link between a woman's sexual interest and a man's smell is even more complex than this. Salty water from a man's sweat glands blends with androstenone from the apocrine glands, and together they nurture the bacteria that live on the skin. It's a microscopic breeding ground that gives every man a unique smell.
Short, sweet little thing. Okay, so that's kind of interesting for some people, I guess. But just keep that in mind that guys usually stink to women.
So don't take a shower when she's smelling
Yeah.
blood or smelling.
So in document sharing, there is a take-home clinical questions for the muscle and the nervous system. I just put that on there today. So work on that over the weekend. Bring it in on Monday. Okay? Questions over the clinical questions. That'll take care of your muscle lab, muscle test. Bring that tomorrow. I'm going to get them all taken care of.
Clinical questions for Monday, you said?
Clinical questions for Monday and document sharing. Yep. Over muscles and nerves.
Alright.